In December of 2025, the USDA launched a $700 million regenerative pilot program through EQIP and CSP, the first federal conservation program that requires standardized soil health testing to verify outcomes. It signals key changes that these cost-share programs are going to be going through. If you've worked with EQIP before, you know how it typically goes. You apply for a specific practice, cover crops, a grassed waterway, maybe a nutrient management plan. You implement it and you get a check. The regenerative pilot changes the structure. Instead of applying for individual practices, you're building a whole farm conservation plan. You can bundle cover crops, reduced tillage, crop rotation, nutrient management plans, whatever fits into your specific operations context, and you fit it all into a single application. That's new, but the bigger shift is what happens after. You're required to test your soil health in the first and last year of your contract. Basically, you need to create a baseline and then a final assessment, and you're still getting paid to implement the practices, but now there's a measurement layer on top. The NRCS wants to see whether the practices actually moved the needle. Rather than being practice-based, now it's outcomes-based. So what does soil health testing actually look like under this program? It's not your standard fertility panel. NRCS is measuring biology, microbial respiration, the nitrogen your microbes are actively cycling, and the composition of your microbial community. It's the stuff that tells you whether your soil is functioning as a living system, not just what nutrients are sitting in your soil. Now they've standardized all of this under a framework called CEMA 216, CEMA. 6 indicators applied to every contract in this regenerative pilot program. In this video, we're going to break down exactly what CEMA 216 is, what the 6 core indicators actually measure, how the NRCS scores them through a system called SHAPE, and what all of it means for your operation. CEMA stands for Conservation, Evaluation, and Monitoring Activity. The 216 is the specific code for soil health testing. This is the NRCS line item that authorizes and pays for soil health analysis under EQIP, CSP, and the regenerative pilot. Think of CEMA 216 as the government's answer to a simple question. How do we measure whether conservation practices are actually improving soil? CEMA 216 changes that. It creates a standardized set of indicators that every participating operation gets tested against. And here's the part that makes CEMA 216 different from running any soil test. The indicators aren't chosen arbitrarily. They were selected because they respond to management changes within a contract period. They're reproducible across labs, and they cover the 3 pillars of soil health: biological, physical, and chemical. There are 6 indicators tested in CEMA 216, and now we're going to go through each one. These are the 6 indicators that the NRCS has standardized under CEMA 216. Each one is measuring something specific about your soil, and together they give you a picture of how your soil functions as a living system. The first indicator is soil organic carbon. This is the anchor indicator. Soil organic carbon tells you the total amount of carbon stored in your soil, not just the active fraction, not the stable humus, everything. The dry combustion method to get this indicator value heats your sample to around 900 degrees Celsius and measures the CO2 released. It's the most accurate method we have for total carbon. But why does the NRCS lead with this indicator? Because soil organic carbon is the single best integrator of soil health. It directly drives water holding capacity, nutrient cycling, microbial habitat, and aggregate stability. When organic carbon goes up, almost everything else follows. When it goes down, the soil loses function. For the regenerative pilot specifically, organic carbon is the indicator that tells the NRCS whether your system is building or depleting. A 5-year CSP contract that shows a measurable increase in soil organic carbon is the strongest possible proof that the practices are working. It's the number that anchors this whole assessment. The second indicator is wet macro aggregate stability. This is the physical structure indicator. Wet macro aggregate stability measures how well your soil holds together when it gets wet. The test takes a dry soil sample, places it on a sieve, submerges it in water, and the percentage of soil that remains intact as water-stable aggregates is your score. This matters for 2 reasons that go way beyond the lab. First, aggregate stability is the primary control of water infiltration. Soils with strong aggregates absorb rainfall, and soils with weak aggregates seal over and shed water as runoff. If you're in a watershed program or anywhere near a drinking water source, this is the indicator that connects your field to downstream water quality. Second, aggregate stability responds directly to tillage and biological activity. Fungal hyphae and root exudates are what glue aggregates together. When you reduce tillage and increase living roots through cover crops, aggregate stability improves. So this indicator is a direct readout of whether your physical management is building or taking away your soil's structure. For operations in the southeast, where we are, where heavy clay soils and intense rainfall are the norm, aggregate stability is arguably the most important operationally relevant indicator on this list. A high score means your soil is handling rain events. A low score means you're losing topsoil every time it storms. The third indicator is just soil pH. But now, why is pH on this list? Because it controls almost everything else. Nutrient availability, microbial community composition, and the effectiveness of your amendments are all dependent on pH. If your pH is below 5.5, aluminum toxicity starts shutting down root growth and microbial activity. If it's above 7.5, phosphorus and micronutrients start becoming less and less available. The biological indicators, soil respiration, nitrogen cycling, microbial diversity, all function within a pH window. If you're outside that window, improving the other indicators becomes much more difficult. For EQIP and CSP contracts, pH also connects directly to nutrient management. If your operation is applying lime or sulfur to correct pH, CEMA 216 gives you a standardized before and after measurement that holds up across sampling dates. Now, the fourth indicator is soil respiration. Soil respiration is a biological activity indicator. It takes dried soil sample, re-wets it, incubates it for 1 to 4 days, and then measures the CO2 that's released. That CO2 is coming directly from your microbial community, metabolizing carbon. More CO2 means more biological activity. It's essentially a fitness test for your soil's biology. This is one of the most responsive indicators to management changes. When you add cover crops, reduce tillage, or increase organic matter inputs, microbial activity ramps up quickly. You can see movement and respiration within 1 to 2 seasons, well before total organic carbon or aggregate stability shows a measurable shift. This makes it a leading indicator. It tells you early whether your practices are feeding the biology or starving it. Although one thing to understand is that higher respiration isn't always better on its own. A field that just got tilled can spike respiration because the biology is burning through a sudden pulse of exposed organic matter. That's high activity, but your soil's losing carbon, not building it. Respiration is most useful when you read it alongside all of these other indicators, especially organic carbon. If both are going up, your system is building. But if respiration is high and organic carbon is flat or dropping, you're cycling through a shrinking pool of carbon. The 1 to 4 day incubation window is also deliberate. It's long enough to capture the response of the active microbial community, but short enough that you're not measuring the slow decomposition of stable organic matter. You're measuring the population that's awake and working right now. That's the biology that's cycling your nutrients, building your aggregates, and suppressing pathogens. For the regenerative pilot, respiration is the indicator that gives you the fastest feedback loop. If you're in year 1 of a 5-year contract and your respiration numbers are climbing, then you know that the system is responding to your practices. If they're flat or declining, you may need to adjust before your final assessment. All right, the 5th indicator we're measuring on the CEMA 216 is bioavailable nitrogen using the ACE protein method. This indicator answers a question that standard soil tests may miss. How much organic nitrogen is your biology capable of making available to your crops? The ACE protein method, which stands for autoclaved citrate extractable, it measures the pool of soil protein that's available for your biology to mineralize into plant available nitrogen. Now here's why this matters. Traditional soil nitrogen tests measure what's already in inorganic form, nitrate and ammonium. But in a healthy, biologically active soil, a significant amount of nitrogen cycling happens through organic pathways. Microbes break down proteins and amino acids, releasing them as nitrogen as a byproduct. That biological nitrogen pool can be enormous, but it doesn't show up on a standard test. The ACE protein method quantifies that pool. It tells you how much nitrogen your soil biology has stored up and is available to cycle. Now, there's a reason the NRCS chose this indicator over dozens of other options. There's a 2026 study that was just released. It was a national effort collecting standardized soil health data across the country. And it tested which indicators consistently separated good management from poor management regardless of the soil type or region. And the ACE protein was one of the 2 indicators that held up everywhere they tested. It's not just theoretically useful, the data says it works, and if you're building soil health, cover crops, and reduce tillage, this number should be growing over time. And as it grows, this is the important part, the amount of synthetic nitrogen you need to apply may shrink. That's the potential economic benefit. A larger biological nitrogen pool means less reliance on purchased inputs. The science connecting a specific ACE protein value to a specific rate reduction isn't fully standardized yet. Nobody's handing you a calculator that says your ACE protein is X, cut 40 pounds of N. But the direction is clear, that as that number increases, the amount that you have to apply goes down. We just don't know exactly what that correlation is yet. But that brings a bridge between soil health data and fertility decisions. For nutrient management plans, especially if you're doing a 590 under EQIP, this ACE protein gives you a data point that a standard fertility test can't. It tells you what your biology is contributing to the nitrogen economy of your soil, not just what's sitting in inorganic form right now. That's the kind of information that makes a nutrient management plan smarter over time. It reveals the full organic nitrogen cycling pool. All right, now the 6th CEMA 216 indicator is microbial diversity measured by PLFA, the phospholipid fatty acid analysis. If the other indicators tell you what your soil's doing, PLFA tells you who's doing it. PLFA extracts those fatty acids and identifies the relative abundance of gram-positive bacteria, gram-negative bacteria, actinomycetes, saprophytic fungi, arbuscular mycorrhizal fungi, and protozoa. We did a full deep dive on PLFA interpretation in our previous video, so I won't repeat all of that here. But in the context of CEMA 216, here's what's most important. PLFA is the indicator that shows whether your microbial community is shifting in response to management practices, specifically the fungal to bacterial ratio. In tilled conventional systems, that ratio is heavily bacteria dominated. As you reduce disturbance and increase living root diversity, fungal biomass grows. That shift towards fungal dominance correlates with more stable carbon storage, better aggregation, and more efficient nutrient cycling through mycorrhizal networks. PLFA also includes stress biomarkers. There are certain fatty acid ratios that shift when microbial communities are under environmental pressure from drought, compaction, or chemical inputs. Those stress markers can flag problems a full season before they show up in your yield or in other indicators you're testing. It's the early warning system for your biology. At $55 to $85 per sample, PLFA is the most expensive single indicator on the CEMA 216 list, but it's also the one that gives you the diagnostic depth to explain why the other numbers are moving the way they are. And it's reimbursable under this program. All right, so you've run the tests, you have the numbers for organic carbon, aggregate stability, pH, respiration, bioavailable nitrogen, and your microbial diversity. Now, how does NRCS turn those raw numbers into something meaningful? That's where this idea of SHAPE comes in. SHAPE stands for Soil Health Assessment, Protocol, and Evaluation. It was developed through a collaboration between USDA, ARS, Cornell, and the University of Missouri. The same research groups that built the earlier scoring systems of SMAF and the Comprehensive Assessment of Soil Health, or CASH, which came out of Cornell. The problem with those earlier frameworks was that they were built on relatively small data sets, and their scoring curves were never validated at a nationwide scale. A score that meant something in upstate New York didn't necessarily mean the same thing in central Missouri or in South Texas. SHAPE was designed to fix that. The original version, published by Nunez and colleagues in 2021, was built on over 14,000 soil organic carbon observations from across the continental US. Compiled from the Cornell Soil Health Lab, the Kellogg Soil Survey Lab, and published research. In 2024, scoring curves were expanded beyond organic carbon to cover aggregate stability, respiration, ACE protein, and other indicators. And in 2025, Vayum and colleagues upgraded the model again, version 1.0S, adding spatially explicit uncertainty so the scoring curves are tighter in regions with more data and wider where observations were more sparse. The system is still being refined, but the foundation under it is substantial. Here's how it works. Instead of comparing your numbers to a fixed reference, like saying organic carbon above 3% is good, SHAPE uses what's called a Bayesian percentile model. It compares your results to a peer group of soils with a similar texture, similar climate, in the same soil suborder. That means a sandy loam in South Georgia is being compared to a sandy loam in similar conditions, not to a silt loam in Iowa. Your score is a percentile from 0 to 100, and it tells you where you rank relative to your peers. If your respiration score puts you at a 95th percentile, that means your biological activity is higher than 95% of soils in your peer group. That's a meaningful comparison. It accounts for the fact that a clay soil in the Piedmont should not be held to the same absolute standards as a prairie mollisol. This is a big deal for operations in the Southeast and other regions that have had historically low scores on national soil health benchmarks. SHAPE doesn't penalize for any starting conditions. It measures whether you're improving relative to soils like yours. And for the regenerative pilot, that's exactly what NRCS is tracking. Are you moving the needle within your specific context? So let's connect all of this to what it actually looks like on the ground. If you're entering into an EQIP or CSP contract under the regenerative pilot, CEMA 216 is your testing framework. You'll need at least a first-year baseline and then a final year assessment. The 6 indicators we just covered are what NRCS is evaluating, and SHAPE is how they'll score it. Practically, that means you want to run the full panel in year 1, all 6 indicators, and that gives you a baseline around biological activity, physical structure, the chemical environment, nutrient cycling, microbial community, and composition. The cost is around $220 per sample, and it's reimbursable through these programs. Then you'd implement the practices, whether it's cover crops, reduced tillage, crop rotations, nutrient management 590 plans, and then you test it again. The respiration and bioavailable nitrogen indicators will respond fastest, usually within a season or 2. The aggregate stability and organic carbon will take longer, but over that 5-year CSP contract, you should see movement if the system is working. The key insight is that the CEMA 216 isn't just a compliance exercise. The indicators are chosen to give you actionable information. If your respiration is climbing, but your aggregate stability is flat, that tells you that your biology is active, but the physical management might be the disrupting structure. If your bioavailable nitrogen is high, but your organic carbon is low, you're cycling through a small pool fast, which isn't sustainable long term. Each indicator cross-references each other. Together, they tell you a story about your soil that no single test can. So how do you actually get these tests run? The lab side is catching up to this standard. Commercial labs are already building packages around CEMA 216. Ward Labs, for example, has a dedicated CEMA 216 package that covers all 6 indicators in one single submission. It's designed specifically to fulfill the NRCS requirements. And under the program, your lab needs to hold a certification through the North American Proficiency Testing Program or an equivalent. NRCS lists the approved certifications in this CEMA 216 document, which will be in the description of this video. And once results come back, the lab or your TSP is going to submit that raw data and your sampling coordinates to the NRCS, and they run it through SHAPE and send you back a scored report. If you're already working with commercial soil health tests, like the Cornell Soil Health Assessment, CASH, then there's significant overlap. CASH measures many of the same indicators: aggregate stability, respiration, active carbon, soil protein. These tests are complementary, not competing. The point is you're not starting from scratch. The lab infrastructure and the testing packages are already there. You're able to go get these tests, send them to your lab, and get these results back and be a part of this program. Now, the operations that are building this kind of data set right now, tracking these 6 indicators year over year across each of their fields, are the ones that will be positioned when outcome-based programs expand, when carbon and ecosystem service markets mature, and when lenders and insurers start asking for soil health data. It's not speculation, it's where the incentive structure is already pointing to. Now, in summary, the federal government just put $700 million behind regenerative agriculture. And for the first time, they're using a standardized soil health testing framework, CEMA 216, to verify the outcomes, not just the practices, the outcomes. We have soil organic carbon, aggregate stability, pH, respiration, bioavailable nitrogen, and microbial diversity, all scored through SHAPE, which is against your soil's peer group. And it's reimbursable at $65 to $220 per sample. If you're still running only a standard nutrient panel, you're making management decisions with a fraction of this information. And you're not positioned for conservation programs, watershed initiatives, and outcome-based markets. The data starts with the test. My name's Jack. I'm the CEO and co-founder of Cultra. At Cultra, we built a platform that helps you organize all of this data. CEMA 216 indicators, PLFA, tissue tests, plant sap tests, standard soil tests, all in one place so you can track the trends across seasons, spot deficiencies, and build the kind of soil health data set that conservation programs and markets are starting to require. If you want to see how it works, the link's in the description. We can hop on a call together. We can get your data uploaded into one collective software. Thank you. I hope you enjoyed this video.